In this video, I'll be showing you a hands-on experiment to explore how photosynthesis works by using leaf discs to measure oxygen production. We'll go through each step, from setting up the materials to creating a vacuum and watching the leaf disc start to float as photosynthesis kicks in. I'll also answer some key questions along the way to help you understand why the leaves rise and what role baking soda plays. The materials I use are transparent cups, permanent marker, baking soda, measuring cup, water, teaspoon, dish soap, light source, single hole puncher, plant leaves, plastic syringe, aluminum foil, paper towels, timer, lab notebook, and pencil. Now, let's prepare the materials. First, we need to find a workspace that can get a little wet. Label 2 cups with baking soda and 2 cups without baking soda. Prepare the baking soda solution by filling one of the with baking soda cups with 300 milliliters room temperature water. Then add about 1/8 teaspoon of baking soda to the water and mix it until the baking soda has dissolved. Add 1 drop of dish soap to the baking soda solution and gently stir until it has dissolved. Avoid foam formation in your cup. To one of the without baking soda cups, add 300 ml of water and 1 drop of dish soap. Set up your light source so it shines straight down onto your workspace. With the hole puncher, cut 20 leaf discs from the plant leaves. Make sure they are all the same size and a complete circle. Avoid punching through major leaf veins. At first, the leaf discs sink to the bottom of the syringe because they still contain air. By removing this air, we can make the disc dense enough to sink completely, which is necessary to observe the effects of photosynthesis in the next steps. The vacuum removes air from inside the leaf discs, causing them to collapse and sink. This simulates an environment where the leaf disc can begin taking in carbon dioxide from the solution, which will allow them to produce oxygen later on and eventually start floating. After we introduce the leaf disc to the baking soda solution and light, photosynthesis kicks in. The discs begin producing oxygen, which collects in the spaces inside the leaf, causing them to float to the surface as more oxygen builds up. The discs start floating because during photosynthesis, oxygen gas is produced as a byproduct. This oxygen fills the spaces inside the leaf tissue, making the discs less dense and allowing them to float to the top of the solution. In the baking soda cup, the discs float faster because baking soda provides carbon dioxide, which is essential for photosynthesis. More carbon dioxide means the leaf discs can produce more oxygen, which helps them float. In the cup without baking soda, there's limited carbon dioxide, so photosynthesis occurs more slowly. In the discs, and the discs take longer to float. The time when half the discs float shows how fast photosynthesis is happening under the given conditions. When the leaf discs produce enough oxygen, they become buoyant enough to float. In cups with more available carbon dioxide, like those with baking soda, the discs tend to float faster, showing a higher rate of photosynthesis.